Okay, so we've got a couple of crosses, or a cross between two genes here, not ones that we study during our lab, but these are fruit fly genes that are affecting the eye color of fruit flies, and one of them is located on the X chromosome. So these abbreviations G and PR, these are Garnet, which is located on the X chromosome, and Purple, which is going to be on chromosome 2. So if these are our parental flies, what were the phenotypes of the parents? Phenotypes for Garnet will be homozygous for G, but what if we have a X-linked G? So now we have to look at this X chromosome as having the G allele on it. And a male, this being the G Garnet recessive allele fly for the male parent, is going to be an XY individual and have only one copy of the Garnet gene. So this is a recessive allele located on the male's X chromosome, and no allele for the Garnet gene located on the Y chromosome. The female parent is not showing the Garnet phenotype, so she's going to have the background wild type allele for the Garnet gene, the G plus allele. And she's going to be homozygous for that. So 2 X's, G plus on both of those. And then she is recessive for the PR purple gene. The male is wild type or dominant for the PR plus allele. And so they have these true breeding genotypes, although just bear in mind that the male true breeding version is not homozygous, but in fact hemizygous. So what kind of gametes can we make? Well, it depends for the male as far as whether he's giving a X chromosome or a Y chromosome. So even though, again, true breeding male, nonetheless still gives X or Y. So only one version of each eye color allele, gene allele, but X and Y is different. So we can set up a Punnett Square. Actually, what we haven't done before is one of these parental crosses that had actually two kinds of gametes produced by the male parent. So female parent here, one kind of X, one kind of PR, male parent, 2 kinds of sex chromosomes and 1 kind of PR. He has the PR plus. If you were to work these out, then of course the X, G plus, X, G, PR plus, PR, X, G plus Y, PR plus PR. Okay, so we produce males or females. They are, you can work out the phenotypes for those. Take a second. So G plus for Garnet means not showing the Garnet phenotype. That's going to be wild type at the G locus. PR plus, at least one of those, means not purple. So it's not Garnet, not purple. It means it's going to have the wild type phenotype. Excuse me. Same thing for the male, G plus and PR plus also has the wild type phenotype. So maybe no difference in the expectation here. Let's see what happens in the reciprocal cross. Oh, I'm sorry, we're working forward to the next generation. So now we have the F1 by F1. Sorry, this isn't terribly well spelled out. These would be the F1 individuals. And just as we solved in the previous slide, 2 kinds of X or Y, 2 kinds of PR gives us 4 kinds of gametes that could be produced. The slash here now is separating alleles for the different gamete combinations. 1, 2, 3, 4 gamete combinations, 1, 2, 3, 4 gamete combinations. Female also 4 kinds of gametes she can make. And just to carry that forward gives us a much bigger Punnett square. And any one of these, you can see that the individuals that result over here are going to get a Y from the father, and this is going to make them male. And the individuals here are getting the X from the male gamete from the father. That makes them female. And then across the top, all of the gametes produced by the female parent are going to have an X chromosome in them. So we can solve for any of these. Let's say we solve for this. Let's go this one. X, G, Y, PR plus, PR plus. So the phenotype here is garnet. And over here, basically not purple. So it's garnet only because it has the recessive allele. It's hemizygous recessive for the G allele. Gives us garnet phenotype there. So I've got this time saver filled in on the next one. And what you see is actually a 9/16 wild type eye color. 3/16 is the purple only. 3/16 is the garnet only. We solve for one of those. And 1/16 is the double mutant. So mutant for the G allele, also mutant for the PR allele. And 9/16, 3/16, 3/16, 1/16, we've seen that before as a result of this dihybrid cross. What's so special here? Well, remember the male and female brackets. The female bracket, if we look at female flies only, we've got three quarters of them showing up with wild type eye color, 1/4 with the purple eye color, none showing garnet or double mutant. Among the males, 3/8 wild type, 3/8 garnet eye color, 1/8 purple eye color only, and then 1/8 double mutant. So difference in the outcome, whether you are a male or female in this case. So some evidence of the sex linkage here. Moving on to the reciprocal cross. Now we've got a purple male and garnet female. And now the male has the G plus allele and then still the Y chromosome. So we get another Punnett square, which is one kind of gamete from the female, two kinds of gametes from the male. Let's see if anything changes here. XG plus XG, PR plus PR, XGY, PR plus PR. This one has a G plus at least one, has a PR plus at least one. That makes it wild type. This one has, oh no, garnet. Darn it, garnet. So this is going to have the garnet eye color. All the males from this cross should have the garnet in the F1 because they are getting the recessive allele from mom for garnet and not getting a garnet allele from dad. So this is one of those first generation evidence of sex linkage that we can see. So these would be the genotypes for our F1 individuals, carrying those forward. Those are our F1 genotypes. And now we can cross those into a big old Punnett square and solve for any of those that we wanted to. So let's solve for this one here. We got X, G, X, G, PR plus PR. So here we got garnet again. And this one, at least one PR plus means not purple eye color. So this is now a female with the garnet phenotype. So let's see if we have any others of that. All right, bunch of garnet phenotypes. And remember, we've got the males and females. Males, the top two rows. I'm sorry, females, the top two rows. Males, the bottom two rows. And same ratios in males and females, but a different ratio than one that we've seen in those autosomal crosses. So this is 3/8 or 6/16 wild type eye color, 3/8 purple, I'm sorry, garnet only eye color. Made perfect sense when I made the orange indicate purple and the purple indicates garnet, right? Two of these, or 1/8, give us the purple only eye color, and 1/8 give us the double mutant eye color. That we don't know what that is, but could be white or something like that. Okay, so in the when you get to this point in your lab, you can revise your hypotheses using a X-linked gene. Whether you have a gene that's X-linked or you might just get a a chance to practice doing Punnett squares on X-linked genes.